Hello, my name is Nicola Lizalde, and I'm going to talk about the coriander germination biostimulated with humic acid. First of all, it is proposed to use a biostimulant to promote the microgreen growth. They improve the productivity and the quality of the plants. Humic acid is found in the sedimentation layers of leonardite and it's formed by chemical and biological decomposition. The molecules of humic acid bind to the plants and help them. Humic acid can increase the plant nitrogen uptake and assimilation. It also regulates the plant and microorganisms' growth and the plant's resistance to stress. It's worth to mention that the half of maximal inhibitory concentration is of 0.6033 nanograms and the lethal dose of 54.8 milligrams per kilogram. These are the steps we follow for the usage of humic acid as a biostimulant. First of all, we selected the biostimulant. We determined the concentration of zero point 0.4 milligrams per milliliter. This was the highest concentration recorded for a similar experiment, so we decided to go for it. We counted the seeds and added 2 grams to 4 trays. Then prepared 100 milliliters of humic acid with a concentration of 0.04 grams. Added it to 3 trays to have the replicates and one without any treatment for the control. Then we put them to germinate, and each 3 days we added water, and each 7 days we counted them and added also biostimulant. Finally, we weighted them and performed the statistical analysis at day 15. For the proliferation test in mammalian cells, we cultured a passage 7 cell line of CACO2 and C2C12 in a 96 well plate. Then we prepared and filtered a 16 micrograms per 4 milliliters of media solution of humic acid. Then we diluted it to 1/5, added it to the cells, incubated them for 72 hours, and then performed an MTS test for proliferation and measured the absorbance of the cells after 1 hour and then after half hour. And finally, we obtained the viability of the cells. For the results of stem length, we obtained an average of 5.27 centimeters for the control and of 5.19 centimeters for the treatment. Then, an ANOVA test was performed to determine if the concentration had an influence over the stem length, and we concluded that the concentration of humic acid does affect the length of the stems. This is also reflected on the average of lengths being the control greater than the treatment. For the germination, we obtain an average of 58.74% in the control and of 51.44% for the treatment. A two-factor ANOVA test was performed to determine if the impact of the concentration and the day influenced the germination percentage, and we concluded that the day has a statistically significant association to the percentage of germination. A Tukey test was performed to know which day was statistically different, and it turned out to be day 15. In the case of the weight, we can clearly see a difference between the control and the treatment, weighing more the control ones than the treatment ones. This is attributed to the fact that some of the plates treated with humic acid got contaminated with fungi. This indicates that rather than enhancing the production of coriander microgreen biomass, humic acid is actually lowering the yields. For the viability test, after taking the absorbance readings and calculating the percentage of viability, we obtained a 98.99% of viability for CACO2 cells and 95.23% for C2C12, which demonstrates that it's non-toxic. In conclusion, this proposal includes biotechnological elements because it uses natural components like biostimulants. It also has potential of scalability, of doing it in vitro to reduce the fungal contamination risk, also exposing other microgreens to humic acid to analyze the respective germination percentage. Finally, even though there exists evidence proving that the humic acid has significant effect on coriander growth, we proved the contrary by noticing more growth in the control trees. It becomes necessary to make some changes to the experimental design.